Hi class, we are still here at Unit 4 on nucleic acids and this is already 4.3 specifically on the flow of genetic information. We are already at the last learning objective of our Unit 4 on the illustration of the biological function of the nucleic acids in relation to replication, transcription, and translation. Flow of genetic information.
What is the central dogma? So the central dogma of molecular biology describes the flow of genetic information from your DNA to RNA to proteins. So replication here is simply the copying of the DNA to form identical daughter molecules. And transcription here from DNA to RNA is the process by which the genetic message from your DNA is transcribed in the form of mRNA to be carried to the ribosome. Then we have translation from your RNA to protein. This is the process by which the message is decoded by the ribosome, where mRNA is used as a template in directing the specific amino acids sequence during protein biosynthesis. So it seems a lot, but this is very simple and it is fun to learn. DNA replication is said to be semi-conservative because each newly synthesized molecule contains one parent template strand and one new daughter strand. So in your book, you can see we have a summary of steps there. We have initiation, elongation, and termination. In the initiation part, we are preparing the DNA template, and we have helicase that unwinds or opens up the DNA, forming a replication fork. So it opens this up, forming replication forks. And multiple replication forks along a DNA molecule create replication bubbles. So this is what you call the replication bubbles. So in elongation, an RNA primase in green here adds a complementary RNA primer in red to each template strand as a starting point for replication. And then DNA polymerase in peach here reads the template strand and adds new complementary nucleotides in a 5' to 3' direction. So this strand under is called the leading strand, and the strand above is called the lagging strand. So the lagging strand, an RNA primer is also laid there, and new nucleotides are added in a 5' to 3' moving away from the replication fork. And the DNA produced here is called an Okazaki fragment. So for termination, a different type of DNA polymerase removes the RNA primer in red and replaces it with DNA in the lagging strand. DNA ligase joins two Okazaki fragments with phosphodiester bonds to produce a continuous chain. And then each new DNA molecule is rewound by helicase. So using the following DNA sequence, what would be the corresponding DNA strand? So remember that in DNA, adenine pairs with thymine and guanine pairs with cytosine. So adenine, thymine, adenine, guanine, cytosine, thymine, guanine, thymine, cytosine, adenine, cytosine, and thymine. Now we go to transcription. Transcription is how RNA is made. Just as DNA polymerase makes new DNA, a similar enzyme called RNA polymerase makes new RNA. So in our book, We also listed the summary of steps for the transcription process. We have initiation, elongation, and termination again. And for the initiation part, RNA polymerase binds to a promoter site and a region of bases that signals the beginning of a gene. And this RNA polymerase is bound to the Tata box of the promoter by transcription factors. Then the double helix, the DNA double helix, unwinds and is ready to be transcribed. On the elongation part, RNA polymerase moves along the protein encoding gene, adding new RNA nucleotides in the 5' to 3' direction and complementary to the DNA template. So unexposed thymine on the DNA strand hooks up with an RNA nucleotide with an adenine. Unexposed cytosine on the DNA hooks up with an RNA nucleotide with a guanine base. But unexposed adenine DNA will hook up with uracil. So don't forget, in RNA, instead of thymine, we have a base pair for adenine, which is uracil. As the RNA polymerase moves along, it already makes the strand of RNA, which is our mRNA, the one in red in this animation. So it is called messenger RNA because it's a messenger. It carries DNA's message out of the nucleus and into the cytoplasm. And remember, mRNA is single-stranded. So what happens next? RNA polymerase reaches the terminator region of the protein encoding gene. And then when it reached the terminator region, all of the enzymes and factors are released, and the product of these three steps is called the immature or pre-mRNA. And the pre-mRNA is processed to produce the mature mRNA, which exits the nucleus and is translated in the cytoplasm. When the RNA polymerase is done reading the gene in the DNA, it leaves. The separated DNA strands reconnect and ready to be read again when necessary. And then your mRNA moves out already of the nucleus and finds a ribosome in the process called translation. So let's look at the next process. Before we go to the translation process, we first check if we still can comprehend with this central dogma of molecular biology. So we look at this question and try it out. Ang tagal lumabas ng question. So using the following DNA sequence, what would be the corresponding mRNA strand? So that means this is the DNA already that will be read by the RNA polymerase, and we will be hooking up the corresponding mRNA strand. So let's look at that. Remember, adenine pairs up with uracil and guanine pairs up with cytosine. So instead of for thymine, that is adenine. For adenine, that is uracil. For cytosine, that is guanine. Cytosine, guanine. Thymine here, adenine, guanine. Cytosine again. Then thymine, adenine. Then guanine, uracil, guanine, and adenine. So that's it. That's your mRNA strand. On the last step of the central dogma, the translation process, the language of nucleic acids or nucleic acid is translated into the language of proteins. That's why it's called translation. It translates nucleic acids to proteins. So before we go to the steps for translation, we look at the key players first. We have the three types of RNA: mRNA, rRNA, and tRNA. We've seen this mRNA from the previous process, our transcription, but we will look deeper on what rRNA is and tRNA. So this mRNA strand that is synthesized in transcription process is composed of codons. And what are codons? Codons are three base sequences of mRNA. So you will see here in our slide, we have what you call the start codon, which is usually A, U, G, and this codes for the amino acid methionine. And then we have stop codons, which is wa, wag, uga, that is U, A, wa, UAG, wag, and uga. Okay, so all others here, all three base sequences are codons. This is a codon, this is a codon, and this is a codon, and this is a codon as well. And then we have an rRNA, or ribosomal RNA. It has two subunits. We have a larger subunit and a smaller subunit. It has two major sites. It has a P site and an A site. So the P site here holds the growing polypeptide chain, and the A site here is where the new amino acids enter. And then we have our tRNA, or our transfer RNA. It transfers the amino acid to the ribosome, or it carries the amino acid to the ribosome. So if we look at our slide, it has a three base anticodon pairs with the mRNA codon. So we have here, we call an anticodon. So during tRNA charging, each tRNA picks up an amino acid from the cytoplasm. We have this another tool for our biochemistry, the genetic code, which shows us the corresponding amino acid for every codon. So if three RNA bases code for an amino acid, that would be 3, and we have 4 nitrogenous bases, uracil, cytosine, adenine, and guanine. Therefore, we have 64 amino acids. But we don't have 64 amino acids. We only have 20 amino acids. So that's more than enough coding capacity. Therefore, we will see here in our genetic code that code is redundant for most amino acids. Just like for valine, we already have 4 here, 4 codons that can code for valine only. And then we have alanine also that can code for 4. So to make life easy, I just really deliberately copy-pasted those parts in the summary in your book so that it's easy for you to remember the parts of the translation process. So again, we have 3 steps here: initiation, elongation, and termination. For the initiation part, the 5' G cap of our mRNA binds to the ribosome. So you see here your ribosome, we have the smaller subunit and the larger subunit. Then the start codon, AUG here, and the anticodon with methionine. This is the anticodon and has a methionine amino acid with it binds at the P site. So you will see that the A site is open and ready to receive new tRNAs. So in elongation, tRNA enters the A site carrying the amino acid, and the peptide bond is formed between the amino acids. So there's what we call translocation. The ribosome moves along the mRNA. Your ribosome moves, and the amino acyl tRNA shifts from the A site to the P site. From the A site to the P site. And then we go to termination. This is when a stop codon is reached. We have wa, wag, uga, and all other parts are released at this part. So let's check again our understanding. Let's try this question. Using the following mRNA sequence, what would be the corresponding anticodon and the polypeptide chain? So this is already an mRNA. So these are codons. So we are looking at anticodon first. So that gives us uracil, and uracil pairs with adenine. And then we have guanine pairs with cytosine and the rest. A, C, U. So these are our anticodon. Then it's also looking for the polypeptide chain. So that would bring us to the genetic code. We have here, we start with AUG. This is the first base, A, and then U. So we stay here, and then G. So that is AUG is methionine, which is our start codon. Let's go to the next one, GAC. G, A, and C, that is ASP or aspartame. And then we have CAG. C, A, and G. And we have glutamine. And then we have UGA. U, G, and A. And that is a stop codon. So we have methionine, aspartic acid, and glutamine. And then we stop there. So what we learned from this lesson is we look at the biological functions of our nucleic acids in the flow of genetic information from replication, transcription, and translation. So we will try this last question as a summary for everything for this lesson. Using the following DNA sequence, what would be the corresponding polypeptide chain? So we start with DNA, and we want to have your polypeptide chain. So what if we replicated first, replication, and then we have transcribe, transcription, and we have translation, so we can get your polypeptide chain. So for DNA replication, we will have another daughter strand for this one. In this case, I want to replicate this again. Okay, so we have adenine binds to thymine, thymine binds to adenine, and then guanine binds to cytosine. And then I will do the rest. Okay, so in the process of transcription, we will transcribe this DNA code here in our mRNA. So adenine will still be paired to thymine here from the DNA, and adenine in our mRNA will bind to uracil already. And then cytosine still with guanine, and I'll do the rest again. You can post this so that you can try it at home. And then we will look at the polypeptide chain already. For translation, we will already translate it to the polypeptide chain, to your proteins. So AUG is methionine, UGA is your stop codon, and GCCAG, we saw that a while ago. We can check that in our genetic code. This is aspartic acid and your glutamine. So that's it. You have your replication, transcription, and translation. See you in our next video.
For activity 4.3.1, you are given a part of DNA undergoing replication, so you need to copy and write the corresponding bases in the new strands, and you need to put an arrowhead in the appropriate end of each new strand to illustrate the direction of the growth. For activity 4.3.2, you're asked to differentiate replication and transcription, and also write the sequence of the RNA transcribed from the following DNA. And lastly, for activity 4.3.3, you are given the mRNA, and you will use this genetic code to predict the amino acid sequence encoded by the mRNA. And the information in this mRNA came from the DNA, so you are asked to give the sequence of the sense strand of the DNA. Lastly, referring to the mRNA here, a TRNA must interact with the mRNA codon CUG, which codes for leucine, and you are asked what will be the anticodon for the tRNA leucine. So what have we learned from this unit 4.3? So first is we've learned about the flow of genetic information, and we also learned about how replication, transcription, and translation happen. So that's it for unit 4.3. Thanks for watching.